Well, thanks, Mike, and I want to thank Mike in particular, and Mort, and the entire Goldberg family, and all of you for being here. It's really humbling and awe-inspiring for me, who's been working on ryanodine receptors since the late 1980s when I cloned the gene, to be able to meet the patients and families, and makes me understand every single day why it's so important that we try as hard as we can to understand how the channel works and to come up with a treatment. I'm going to talk the next 20 minutes or so about a new class of drugs that we call Rycals, discovered in my laboratory in the last 10 years. And this is an example of a Rycal. They are very simple molecules, small in size. The ones that we work with primarily can be dissolved in water, so they can be taken as a pill orally. And as I'll show you, they fix this leak in the ryanodine receptor that you heard from Bob's beautiful talk a moment ago. And I'll talk to you a little bit about how they do that, what the leak is, and what we're planning to do with them. So the ryanodine receptor, as you've seen, is a channel, meaning that it makes a hole in a membrane that allows calcium to pass through a protective channel, and that calcium, as you heard from Bob, is what makes your muscles contract in both your limbs and in your heart. And over the years, we've done quite a lot of work to understand how the ryanodine receptor, abbreviated RyR, works. It turns out it's extremely complicated. It's the largest known ion channel. Unlike most other ion channels, it's located inside the cell. So a lot of the standard techniques that one uses to study ion channels cannot be used on ryanodine receptors. And it's present in virtually all types of tissues. But in particular, we've studied it quite a lot in skeletal muscle and in heart, but also in brain. The form that's in the skeletal muscle that's involved in the RyR1 myopathy, obviously, is RyR1. The one in the heart predominantly is called RyR2. Essentially, the ryanodine receptors control the flux of calcium, as you heard from Bob. And when these channels open, calcium floods into the cell, and this makes your muscles contract. And when it closes, your muscles are able to relax. In certain disease states, and I'll show you more about that in a moment, ryanodine receptors can become defective. And one of the types of defects that we and others have studied is what we call a leaky defect in the channel. And this means that the channels don't close properly. And this can have a number of different consequences depending on which organ the leaky channels are in. And we've studied them not only in skeletal muscle, where they can contribute to muscle weakness, but also in the heart, where they can cause arrhythmias and heart failure, and even in the brain, where they've been linked to post-traumatic stress disorder, Alzheimer's disease, and Huntington's disease. We know what causes the leak in both individuals who have mutations in the channel. The mutations can cause a leak, but also in individuals with quote-unquote normal channels, but other associated diseases, such as heart failure, diabetes, cancer, or simply normal aging, where metabolic abnormalities such as oxidative overload can cause the channels to become leaky. And quite remarkably, and I got asked this question yesterday in our scientific advisory board meeting, the identical same type of leak that occurs in ryanodine receptors in RyR1 with mutations in the channel also occurs in RyR1 in patients with Duchenne muscular dystrophy. So even though their ryanodine receptors are genetically normal, the oxidative stress of their disorder caused by a completely unrelated gene also causes the same type of leaky ryanodine receptors. And this explains why the strategy that we're taking in partnership with Servier and Armgo is to use the same type of Rycal drug that fixes this leak to treat both boys with Duchenne muscular dystrophy and individuals with RyR1 myopathy. The mechanism by which the drug works is to prevent the loss of a stabilizing subunit on the channel that we call calstabin. And so under stress conditions, or if you have a mutation in your channel, this calstabin doesn't bind tightly to the channel, and the channel becomes leaky. And the Rycal drugs prevent the loss of calstabin. You can think about this like the screen door on your house and the spring on the screen door. If that spring comes loose, the door is going to be flapping open in the breeze, and the cats and dogs and kids will run out. You put the screen door, the spring back on, that's the calstabin. You can still open the door when you need to, so the channel can open normally, but it stays closed when you don't want everybody running out. And that's kind of what the calstabin does. Well, everyone in this room knows the word ryanodine, but I bet very few people in the room know where that word came from. And this is where it comes from. This is the Ryannia speciosa plant that grows in South America. And natives, taking their blow darts to shoot at their enemies, put a little bit of extract from this leaf on the tip of their blow darts because it contains a chemical called ryanodine. And when ryanodine gets into your body, it binds to your ryanodine receptor channel and locks it open. And you can imagine what happens if the channel is locked open. Your heart turns to stone, your muscles can no longer relax, everything is just contracted, and you die. And so these natives in South America figured out that they could use this plant extract to kill their enemies. The plant has it because when an insect lands on this leaf and takes a bite, it also gets paralyzed and falls off. So DuPont has taken advantage of this and made a derivative of ryanodine as the most potent insecticide ever developed commercially, 1,000-fold more potent than DTT, and presumably specific for the insect cell ryanodine receptor, not affecting mammals. So Bob gave a beautiful review of how muscle contraction works, and when a muscle cell's electrically activated, a little bit of calcium comes in, binds to the yellow ryanodine receptor, which opens and allows calcium to be released from this intracellular store, binds to the sarcomere, causing shortening, and then relaxation occurs when that same calcium gets pumped back in to that organelle inside the cell. So every time your heart beats, every time you move a limb, this cycle of calcium release through the ryanodine receptor and reuptake occurs. And again, one of the defects that we have found associated with RyR1 myopathy is a problem with the channel not closing tightly enough, so that when the muscle's supposed to be relaxing, instead of the channel being tightly closed, the muscle being relaxed, it continues to leak calcium out and the muscle can't relax properly. If you think about it, when I move my arm, the muscles on the inside of my arm have open ryanodine receptors and they're contracting. On the outside of my arm, the muscles have closed ryanodine receptors and they're relaxing. If those ryanodine receptors were leaky, I would have trouble just simply moving my arm or walking or breathing. So over the last 15 years or so, we have found evidence of leaky ryanodine receptors, either RyR1 or the closely related RyR2, in all these different organs: brain, heart, skeletal muscle, pancreas. And these are some examples of papers we've published showing that if you have a leaky ranitidine receptor in the brain, for example, you can get seizures and cognitive dysfunction. In the heart, it can contribute to heart failure and arrhythmias. In skeletal muscle, it can contribute to muscle weakness in muscular dystrophy, but also simply age-related loss of muscle function. And even in models of human breast cancer, metastatic to bone, ranitidine receptors become leaky in skeletal muscle and explain why patients with cancer have muscle weakness. And we have quite a lot of data now showing that muscle biopsies that we've gotten from patients with RyR1 myopathy, some in this room, as Mike had mentioned, have also leaky ranitidine receptors. And even in the pancreas, in the cells that make insulin, they can have leaky ranitidine receptors that can cause diabetes. And this table summarizes, again, some of these findings, listing some of the diseases, both common ones like heart failure, arrhythmias. Sarcopenia simply means age-related loss of muscle function, but also other rare genetic diseases like RyR1 myopathy, but in this case, CPVT, which affects the heart, causing arrhythmias. These individuals also have leaky ranitidine receptors, the RyR2 type. And so the idea then that's emerged from many, many studies is that in the case of a ranitidine receptor that either has a mutation that prevents the tight binding of the stabilizing subunit or has been subjected to stress conditions like oxidation or both, the stabilizing subunit, the spring on the screen door, cannot bind the channel tightly. The channel opens normally, just like a normal channel. The defect is that it fails to close properly. And that is the therapeutic target of the Rycal drugs, to prevent this pathologic leak of calcium and restore the channel to the normal, non-leaky condition. So over the last decade or so, we have conducted many, many studies in animal models showing that the Rycal drugs, which bind to the ranitidine receptor, but do not block the pore of the channel, they merely prevent the loss of the stabilizing subunit, and they've been effective in preventing heart failure progression. They prevent arrhythmias and improve exercise capacity in animal models. And with Servier, we've gone on to do early phase 2A clinical trials with one of the Rycals in patients with heart failure. And there was a pharmacologic effect where they improved the function of the heart, measured as ejection fraction, reduced the left ventricular size, decreased brain natriuretic peptide levels in this clinical trial. The trial was a short duration. The dose-dependent effects were not very strong, and the drug that was used in this early trial was not very potent. And so the decision was made not to advance that particular Rycal, but to move on and develop more potent second-generation Rycals, one of which you'll hear about is ARM-210, which in September 2015 started phase 1 safety trials. Now, one of the closely related drugs that we have used in a lot of the animal studies is called ARM-107, or S107. And we have shown that in these animal models of muscular dystrophy, that the ranitidine receptors undergo remodeling, principally due to oxidation. They lose the stabilizing protein, and this applies not only in the mouse models of muscular dystrophy, but also in our unpublished work, we've shown this for the RyR1 myopathies, and that these Rycals prevent the depletion of the stabilizing subunit, decrease evidence of intracellular calcium leak, which we measure as something called sparks, and improve muscle function, decrease the CK leak, and improve the histology of the muscle. And functionally, the Rycals have improved exercise capacity, grip strength, and other functions of a muscle performance. Without getting too technical, one of the key ideas in muscle is that when calcium is released to make muscles contract, it's done in a very controlled fashion. Whereas in the case of an RyR myopathy mutation, that release of calcium is not regulated adequately, and there can be pathological consequences of that abnormal leak. And that can include depletion of calcium in the pool. So like if you stick a screwdriver in your gas tank, the gas leaks out, you go to put your foot on the pedal, as we discussed earlier, no oomph, that's part of the problem. But also that leak of calcium can itself be damaging to muscle. So we do lots and lots of animal studies, and we can get a lot of detailed data about the performance of muscle. So for example, we can measure the force production in the muscle that we isolate from the mice treated with a Rycal and show in red that it's improved. We can ask the mice to grip onto a rod, and we can see how strong their grip strength is and show that that's improved. We can also look at the histology of the muscle coming from the mice that have been treated and show that the muscle fiber size is improved and that there's evidence of less intracellular calcium leak. I show examples of these to tell you how powerful the tools are that we have in all the labs of the people here, the scientists here can do these types of studies to really ask at a very detailed level both what the defect is and what the drugs can do to fix it. But importantly, all of this is designed to come up with a treatment. And the drug that we're focusing on now is called ARM-210, or S48168, which is our partner Servier's number. And some of its characteristics are shown here. It's been very well characterized so far. As I mentioned, it can be taken orally. It's a small molecule, and it has good activity on its primary target. It's similar to, but distinct from the other Rycals that we've tested. And it has a very good tissue penetration in skeletal muscle and a longer half-life compared to the ARM-036 that was used in the heart failure clinical trial. It seems to have a safe pharmacologic profile, and so far, no safety alerts. We heard yesterday in a summary from Supan Shah, who's the CEO of ARMGO, that it has been tested in several laboratories around the world in the Duchenne muscular dystrophy MDX mouse model and shown to have good activity. And it's patented. So there have been a number of studies done using this Duchenne muscular dystrophy mouse model. And the mouse model is very simple. You put the mice in a cage, you put the drug in the drinking water, and mice love to jump on a running wheel, and you record how many revolutions the wheel makes, all by computer, and get a very, very accurate measure of how much exercise they're doing. And so using that type of simple test, for example, we and others now have shown that normal mice run about 8 to 10 kilometers a night, and the dystrophic mice about 4 to 5. And when you treat the dystrophic mice with the Rycal, it significantly improves their exercise capacity. We can also take the muscle out and directly measure the strength of that muscle, and that's also significantly improved. So where do we stand now with ARM-210? As I mentioned, it's got a favorable profile for development as a drug. We've established a minimum effective dose in the MDX mice. The phase 1 safety trial started in September 2015, and if all goes well, the plan now is to start phase 2 in Duchenne muscular dystrophy, which will be headed by Servier. And we are in planning stages for an RyR1 myopathy trial, hopefully to start in 2017, which would be performed at the NIH, directed by Katie Milieu with ARMGO. Now, designing clinical trials is extremely challenging, and here's the model that we've come up with so far. A lot of details still to be worked out, but we are very hopeful to get this trial going as soon as possible. And I thank you all for listening, and be glad to take questions.
Thank you. That was excellent. Thank you so much. Any questions? JP?
Hi, hi. I think you said that, very exciting, by the way, very much looking forward to seeing that available, that your drug effectively helps fix leaky channels, and not so much if you suffer from a narrow channel or a blocked channel. Can you comment on whether most people with an RyR myopathy would suffer from a leaky channel versus a blocked channel, and whether that's more likely to occur in those that suffer from a dominant mutation versus a recessive mutation?
Yeah, so those are critically important questions, and we have an ongoing project you heard a little bit about yesterday. We're putting together a database of as many patient mutations as we can get a hold of to understand what the natural history of the disorder is based on the type of mutation, and whether or not the Rycal drug would have any benefit in different types of mutations. So we don't have an answer to those questions yet, but I think we're collecting data now at a pretty rapid pace and should have more information in about a year or so.
I was just wondering, when you said that there was a significant improvement in the mice, how significant that was, and whether or not this would be more of looking like a cure or just more of looking like an improvement, and how much of an improvement, if you can even say.
Yeah, that's a great question. I think it's impossible to take a leap from results in mice to how things might look in a human. The effects that we see in the mice, we compared to what is seen with steroids, which is the current standard of treatment in Duchenne muscular dystrophy, and it was comparable. We also compared it to published results from exon skipping trials, pretty much everything that was out there. And the Rycals were as good or better than everything that had been published in different preclinical models. All of that is with a big caveat that there's very little clinical material to go on to compare it to. So I think Bob had mentioned maybe that ultimately a drug like a Rycal could be seen as a combination therapy with other treatments.
Come over here. Hello? Oh, now it works. Okay, I have two questions. My first question is there a genetic connection between the leaky heart valve in a family member and the leaky valves in an RyR1 member?
So the channel in the heart is RyR2,
Right,
and the one in muscle is RyR1.
right.
So an RyR1 myopathy should not have a direct effect.
I think she means like aortic or mitral or a macro, a valvular.
Or even in a family, like if a family member has the RyR1 too,
Oh.
and another family has an RyR1 or RyR1, is there a connection between those two?
No, they're encoded by different genes on different chromosomes, so they
Okay.
don't track together.
Okay, and the other question I had was, once that calcium leaks and it's not being used, where does it go? Does it affect bone calcium? Does it make bones softer? Does it affect teeth, et cetera?
Yeah, that's very excellent question. As far as we know, that calcium, first of all, is at a low amount. If you look at the total body amount of calcium, it can be a lot of calcium if it's inside a cell, because it's a very small space. We presume that the calcium that's excess mostly gets excreted from the cell, and it would be diluted into a vast pool of calcium and not have much of a metabolic effect outside the cell.
Thank you.
Hi.
Hi.
You mentioned that RyR1 is used on mouse models that have Duchenne and other muscular dystrophies. I know that Susan Hamilton has mouse models with our central core disease. Have you been tested these mouse models, and is RyR210 better for us than the RyR1?
Yeah, that's a great question, and something we were talking about yesterday in the Scientific Advisory Board meeting, the need to test additional animal models, mouse models in particular, the CCD models. And we definitely want to do that, and we haven't done it yet.
You haven't done it?
Not yet.
So, and I will just put a plug in here for fundraising. So one of the things I asked the same exact question you asked yesterday. Has the Rycal been tested in the CCD model, the YS mouse that Dr. Hamilton has? And it has not. But we want to test this on multiple different mouse models with different mutations, but each of those mouse models costs around $20,000 to $30,000 per mouse. And there's no guarantee that when you pay that money, you'll get the mouse back. It may not work. So we want to try and do multiple, sort of have a sledgehammer or like a fire hose approach where we're doing multiple mouse models with the hope that some of them will come back and be available for research. But
Now, can we as human beings volunteer to be the mouse models and test it?
You can do that once we get the phase 1
FDA.
study. The safety study has to be completed, and then get FDA approval to do a clinical trial.
all right, so we're a little bit behind. So I just want to move on to our next speaker. But thank you, Dr. Marks, for that great talk.